Hello and welcome to another special edition of EUFOREA TV Breaking News. My name is Dr. David Bull. I'm a medical journalist and I'm here with the latest medical news on the COVID-19 pandemic. Now, nearly 40 million people have now been diagnosed with the disease, leading to over 1 million deaths worldwide. And of course, the search for a vaccine continues apace. But whilst that remains a pipe dream, treatments have improved dramatically, as have patient outcomes. But the big question is, which treatments work and which don't? Well, the world's biggest trial of potential therapies is now being carried out here in the United Kingdom at the University of Oxford. It's called the Recovery Trial, which stands for Randomized Evaluation of COVID-19 Therapy. And here to tell us more about it is Professor Richard Haynes. Now, Richard is Professor of Renal Medicine and Clinical Trials and is the coordinator of the Recovery Trial. Thank you very much indeed for taking the time out to talk to us, Richard. Let me start by just asking you, the scale of this trial is, well, it's quite extraordinary and involves a huge number of patients over many hospital sites. It was also set up in record time, wasn't it?
Yeah, when the pandemic sort of got going in the UK, we did realise that we needed to get things going very quickly. So actually the Chief Medical Officer, Chris Whitty, said that once they approved funding, he wanted the first patient in within 2 weeks. We managed to deliver.
Well, that's extraordinary. So how many patients have you actually managed to enrol in total?
Just shy of 14,000, I think, as of now.
And in terms of the patients that you've recruited, what sort of patients are they? Are they patients who are mildly affected, severely affected, or hospitalised? Who did you include?
So the protocol is deliberately really simple. So to be eligible, you just have to have proven or suspected COVID-19 and have been admitted to a hospital. So there's actually quite a broad range of how ill people are. Some people, about 1 in 6, are on a ventilator at the point that they're recruited, but about a quarter aren't on oxygen at the time they're recruited, but they are ill enough to be in hospital.
Okay, so let me ask you then, so what is the design of the study and which therapies did you look at initially? Because I understand there were many arms to this.
Yeah, so it's the recovery has what's called a platform design. So it gives you the bare bones of a trial, and then you can add drugs in and out as you need to. So when we first started, we were comparing dexamethasone, the steroid. We had hydroxychloroquine, the anti-malarial drug, in the trial, lopinavir, ritonavir, which is the anti-HIV drug. So they were the first 3 drugs we started with, and we were comparing each of them with a control group. Since then, we've added in more drugs. So I could tell you about those, or we could start with those 3. So we then added in azithromycin, an antibiotic, into that. And then we started making the protocol a little bit more complicated. In the next drug we added was tocilizumab, which is a monoclonal antibody against interleukin-6. So it's a very powerful anti-inflammatory drug. But we only added that in for a subset of people who were hypoxic, lacked oxygen in their blood, and were inflamed, according to their blood tests. So only a subset of people could be randomized between that and control. And then more recently, we added in convalescent plasma for all participants, but that was done in a way so they could still get azithromycin or dexamethasone or hydroxychloroquine or whatever it was. They could be randomized there, and then at the same time, they could be randomized between convalescent plasma or not. And then just most recently, in the last week or 2, we've added in the Regeneron monoclonal antibody alongside the convalescent plasma. So people now get the monoclonal antibody, convalescent plasma, or not.
Well, of course, that hit the headlines when Donald Trump was given it, and I was fortunate enough to interview Christos from Regeneron about that. Now, I suppose the beauty of this protocol is it's entirely flexible. So, as you say, you've sort of dropped some arms, you've added others.
Yeah, that's been the beauty of it, and we've been able to do it quite quickly. So as results have become available, like the dexamethasone results where it was effective, or hydroxychloroquine where it wasn't, unfortunately, we've been able to take those arms out. And then when interesting new drugs have come along, like convalescent plasma or the monoclonal, we've been able to get them in there pretty quickly to start getting answers.
Well, so what sort of outcomes are you looking for? You must clearly have a primary outcome and then secondary outcomes.
Yeah, that's right. So the primary outcome for all the drugs, they all share the same outcomes. But the primary outcome is death from any cause within 28 days of randomization. Secondary outcomes include duration of hospitalization, and for the people who weren't on a ventilator when they went into the trial, the need for going onto a ventilator or death. There's a composite outcome there for the secondary.
Okay, so what are the early results showing and indicating?
So the first result that came out of the trial back in June was the hydroxychloroquine results, where we found that unfortunately hydroxychloroquine didn't improve outcomes. So we then removed that from the protocol. That was shortly followed by us. We decided that we'd recruited sufficient people into the dexamethasone comparison. We had 2,000 people on dexamethasone and 4,000 controls. And so we decided to stop that arm. We thought that was satisfactory. And actually, we got some very positive results from that. So we saw that dexamethasone reduced the risk of death amongst people who were on oxygen, or had an even bigger effect for people who were on a ventilator at the point that they got randomized. And then more recently, we stopped the lopinavir ritonavir arm, unfortunately, again, because that didn't show any sign of benefit. So at the moment, we're still investigating azithromycin, tocilizumab, convalescent plasma, and the monoclonal antibody.
And when do you expect full results from those arms?
Yeah, we get that question a lot, and we don't really know, can't give a very precise answer. What we are setting out to do is to get sufficient numbers of people into those arms to get a really reliable answer, which we, as I said, for azithromycin, we think we might need 2,000 people on azithromycin, 4,000 controls to compare them with. And we're over halfway there, but we've still got a way to go possibly on that. For the other comparisons, we're a bit further behind that. So potentially longer to go, but it all depends on how effective these drugs may be, because if they are more effective than we think, then our data monitoring committee, who look at the results at regular intervals, might advise us that we've got compelling evidence, and then we would stop.
So, I mean, clearly this trial is of global significance. I mean, pretty much everyone in the world is sort of pinning their hopes on you. But for you personally, what are your hopes and aspirations?
I think what I'd love to see is this trial to continue to develop really reliable evidence. The dexamethasone result was a once in a career moment, probably, but not for my benefit. But it'd be great if this trial could identify other treatments which have similar effects on mortality, because there isn't going to be one golden bullet for this disease, unfortunately. It's likely we're going to need a series of drugs, all of which have a moderate effect, but then when you put them together, they can add up to quite a big effect. And then the other thing I'd hope is that a lot of the lessons that we've learned from this trial about how they can be run in a really streamlined fashion could be applied to other trials in other diseases. So when we can turn our attention back to those other diseases in trials, that we change the way we do them, given what we've learned from recovery.
Well, I just have to thank you so much, Richard, for such an insightful interview. I mean, I really do wish you the best of luck. I mean, I think everyone, as I said, is pinning their hopes on you. No pressure then, but I wish you the very best of luck with everything.
Thanks very much.
Now, you can find more information, and you can also register for the meetings on the EUFOREA.eu website. And there you can also sign up to receive the latest news via email. You can also follow us on Twitter. The address for that is @EUFOREA. But that is it for this episode of EUFOREA Breaking News. See you soon. Thanks for watching. Bye-bye.